Hello and welcome to our very important discussion today. A segment that is very important in our discussion of neoplasia, and that will be the difference between metastasis and invasion. What are the differences between the process of a tumor metastasis and a tumor invasion? And the best way to understand the differences between a tumor metastasis and a tumor invasion is by going through and unraveling the pathophysiological steps that unfold in every step in invasion versus metastasis. That is the best way to understand the differences between invasion and metastasis. To understand the differences between invasion and metastasis is the most important objective in the study of neoplasia as a whole, because what defines a carcinoma, or what defines cancer, is when we come to the staging as to, is the tumor in situ, is the tumor benign, or is the tumor beginning to invade the basement membrane, or has the tumor metastasized to distant organs. So we will first begin by going through the processes of invasion, because invasion is the prerequisite for metastasis. There will never be metastasis unless there was invasion in the first place. And what are we invading? We are invading the basement membrane. This is very important to understand. We have to understand that the tissue compartment is comprised of cells, and these cells are surrounded by the extracellular membrane, which itself, the extracellular membrane, is divided into components known as the connective tissue or the interstitial tissue, and we also have the basement membrane. So without any further ado, let's go to the diagram. On this side, we will elucidate and go through the steps of invasion. Then we will come back and we will go through the steps of metastasis. Okay, so the first thing is we begin with one cell known as the transformed cell, that one bad apple that turns the entire orchard into the basket into bad apples. So if you examine here for just purposes of intensive purposes of understanding, we have 1, 2, 3, 4, 5 cells, and the transformed cell is the one in the middle. What do we see? We see that all these cells are connected. You see that those pink fence-like lines? These cells are held by junctions, these pink junctions. Now, in order for the tumor cell to start invasion, to become an invasive tumor, it has to first of all cut and break those proteins that hold it together with its neighboring cells. And these proteins are called cadherins. They are not fences. Don't say the fences proteins. They are called cadherins, and they are connected with, these are extracellular proteins that they are connected to intracellular proteins known as beta-catenanes, but that is not our part for now. So these are the cells. The orange part is our basement membrane, and the yellow part is the connective tissue. So in order for the cell to become invasive, it has to break those cadherents, or in other words, these cadherents will become defective and they will never be maintained due to the transformed cell, due to a genetic injury that fails to produce those cadherents. So the genes that make cadherents, when there is neoplasia, they will be defected. So the cell can no longer make those cadherents, so they cannot connect the cell to the nearby cells, so there will no longer be any more cadherents, and that means the cell is now free. The next step, after the cell breaks ties with the neighboring cells, it will start penetrating, invading the basement membrane, this orange part, and then it will move down into the tissue, and then it will go into the bloodstream, and then we will start metastasis. So what do we see here? We see normal cells connected to each other as they should by E-cadherents. E-cadherents, letter E dash cadherents. Now let's look at the next stage. What do we see here? We see, as we said, those cadherents are now defected due to the cancer, due to the genetic defect in the genes that make the proteins cadherents. And now the first step is the cell is now liberated. The transformed cell, the bad apple, is now liberated, is free from its surrounding. Now it's free to move, locomotion. Now let's look at the next stage. By the way, what are these scissor-like, this pathetic attempt of drawing scissors, these scissor-like enzymes represent? These enzymes represent mineral or membranous metalloproteinases or collagenases. What are collagenases important for? You see those blue lines? That's collagen. So now in order for the cell to penetrate and invade the basement membrane, it has to secrete enzymes that degrade and break and scissor and destroy the type 4 collagen that is an integral part, that is a very important component of the cellular basement membrane. It will also release matrix metalloproteinases, and among other enzymes that the cell, the transformed neoplastic cell, will secrete them in order to destroy and break the membrane below. Then what do we see in this third part? You see how the cell moved? The cell has now degraded the orange, degraded the basement membrane, and now it went to the yellow part, the connective tissue, and now the cell is ready to move out into the bloodstream. So that is, in essence, the process of invasion. Now, let's look at metastasis. Let us look at metastasis. What do we see here? Where is our bad apple? It's right there. That's our transformed cell. That is the cancer cell. That is the cell that used to be normal but underwent a genetic defect. And the damage is caused either by ultraviolet radiation, either by the failure of systems that repair misfolded proteins, or systems, intracellular built-in systems that properly remove those misfolded proteins, or maybe there was a reactive oxidative species that damaged the genetic DNA, but for whatever reason, now we have a transformed cell. So this transformed cell will undergo clonal expansion. So the first step is clonal expansion, and then it will reach out for blood vessels. That is angiogenesis. This brown barrier over here, what is that? That is our basement membrane. So now we have seen the transformed cell grew into clonal expansion, and now these cells have, as we talked about in the invasion, broke down the side cadherins and those proteins that connect that cell to nearby cells, broke in, and now we have a metastatic subclone. A metastatic subclone is a transformed cell that successfully invaded the basement membrane and is now liberated to move through the connective tissue, looking for a blood vessel to enter the bloodstream and travel for far sites for metastasis. So the first step is a transformed cell. Second step is clonal expansion. Third step is the successful invasion that will now yield a metastatic subclone, a clone of transformed cell that are now able to, through locomotion, to move into nearby blood vessels. And following the invasion of the basement membrane, those transformed subclones will undergo adhesion. So now they will adhere to what? They will undergo adhesion, they will attach into the basement membrane of blood vessels. So now blood vessels also have basement membrane. So now those transformed cells did not only invade the basement membrane of the tissue, now they will invade the basement membrane of the blood vessels. And when we have, as we said, adhesion will take place after the transformed cells, those metastatic subclones, pass freely through the extracellular matrix, this connective tissue, and successfully adheres into the blood vessels. And after they adhere into the blood vessels, they will start to invade the blood vessels. And when those metastatic subclones, when we have those metastatic cells successfully penetrating and entering the blood vessels, this is now called intravasation. Intravasation. Intravasation is when the transformed metastatic subclones have successfully adhered to the blood vessels, and now they successfully invaded and broke and destroyed the basement membrane, and they have successfully entered the bloodstream. That is intravasation. They will ask you the difference between intravasation and extravasation, and as we will see later. What is that purple cell? What is that purple cell? That purple cell is a lymphocyte. That is an immune system defensive cell. Those metastatic subclones don't immediately think that they will successfully make it through the bloodstream. Some of them, most of the time, they will be destroyed by our immune system lymphocytes. As we see here, that pink or that purple lymphocyte has now attached into that metastatic subclone that has now entered, intravasated into the blood vessel and travel, and it will destroy it and remove it. But in other instances, in other unfortunate instances, we will have the metastatic subclone intravasate into the blood vessel, but now platelets, these yellow platelets, they will attach to it, and now we will have a tumor embolus. Do you think a tumor embolus is now easy to be degraded by lymphocytes? No. So now the tumor is now safe to travel in our bloodstream. Unfortunately, our immune system lymphocytes can never interact with it. As we will see here, that's interaction with host lymphocyte cells. But here, when the tumor cell is resistant and will attach to platelets, now we have a tumor embolus. Now this tumor embolus is very, very difficult to degrade by the immune system. So the tumor embolus will travel, and it will adhere again to the basement membrane from the inside. And when it adheres into the basement membrane, it will start growing and growing until it attaches to the other side, and it will now invade again. It will destroy and break down the basement membrane of the blood vessel and exit. And now that is extravasation. Extravasation is when the transformed tumor cells, those metastatic subclones, have successfully traveled through the bloodstream and have now adhered or adheres to the blood vessel and are now penetrating the blood vessel from inside and invading the basement membrane and trying to exit to a far and a distant organ. And when they make their way out, you will have a metastatic deposit. That metastatic deposit will start releasing growth factors that will recruit blood vessels and encourage angiogenesis. Now these metastatic deposits will link themselves and make for themselves a mini circulation with blood vessels to provide themselves with blood, with nutrition, with water, with oxygen, and with the blood will remove all the metabolic waste and as carbon dioxide and remove it from those tumor cells, so these cells can now survive and unfortunately grow and keep growing and do the damage that they are known to cause. So let us now examine our diagram once again. My apologies for my camera skills. Okay, that's supposed to be like this. What I want you to have in mind, what I want you to have a clear idea of, is that in neoplasia, the most determinant or the most critical, the most important concept that helps identify the prognosis, the diagnosis, that helps in the treatment, is to actually identify metastasis versus invasion. And as we said, invasion is basically when the transformed cell, it begins with one cell, one bad apple, that turns into subclones through clonal expansion. And that cell will now begin to destroy the basement membrane beneath. And you have to also remember the causes that lead to neoplasia, and that is a defect in the genes. So in the cell, there are genes that do one of two things. They either promote cell division and entry into the cell cycle and successfully passing through the cell cycle and dividing. And these are the plus genes. And we have the minus genes. The plus genes are the accelerators, and the minus genes are the brakes. So when there's a defect in the plus genes that lead to promotion of growth and uncontrolled cell division, such as when an oncogene, when a proto-oncogene becomes an oncogene, that's a form of a plus gene defect that leads to cancer. There's also the minus genes, which are, there are built-in systems in the cell that commit apoptosis, promote apoptosis if the cell is damaged beyond salvage, if the cell injury can never be remedied, if the cell will now become a destroyed cell. There are pro-apoptotic proteins like the BCL and those very important pro-apoptotic proteins. If there's a defect in the gene that gives us the pro-apoptotic proteins, then we will have cancer, because the cells will undergo uncontrollable cell division, but the pro-apoptosis genes are defective, so we will never have those counter-regulatory mechanisms that properly help the cell get rid of itself as in apoptosis. There's also other important proteins like RB, retinoblastoma, and there's also the P53. It's very important this p53 anti-tumor protein. If the gene for the p53 is damaged, if the gene for p53 is damaged, it can no longer transcribe for us p53, then there will be unopposed, uncontrollable cell division. After the cell has now transformed, whether by a defect in genes that lead to the promotion of uncontrollable cell division, or by a defect in the genes that lead to inability in transcribing or translating those anti-cancer cancer proteins, we will now have a transformed cell. That transformed cell will start to undergo clonal expansion throughout the basement membrane. Then it will use metalloproteinases, it will use anticollagenases to break down those structural components, those structural components that make the basement membrane. Then they will travel through the extracellular matrix connective tissue and will adhere to the blood vessel basement membrane from the outside. Then they will start again destroying, using those metalloproteinases, those anticollagenases, those proteinases, destroy the basement membrane now of the blood vessel to enter the bloodstream, and that is known as intravasation. When they successfully enter the bloodstream, does not always mean they will survive. Those transformed tumor cells, that doesn't mean they will always survive and make it in the bloodstream. Sometimes the immune system's host defense mechanisms, those lymphocytes, will destroy these tumor cells. But in other instances, such as when platelets aggregate with those tumor cells, it becomes now too overwhelming for the immune system's lymphocytes, and they will travel through the blood, and now they will adhere from inside to the bloodstream, and they will travel very far in the blood stream and go to very far distant sites, and they will now start destroying the basement membrane from the inside and exit the bloodstream, and that is extravasation. And when they exit, they will make this tumor embolus will travel with platelets and will adhere to the basement membrane from inside and extravasate and make a metastatic deposit that will later on secrete pro-angiogenesis growth factors that would recruit promote angiogenesis and bring blood vessels that will enrich this basement membrane from outside. Those metastatic deposits will now have blood, and the blood is the source of life for cells, whether transformed or normal cells. Now these transformed cancer cells will now have a rich source of blood supply. They will have nutrition. They will now have a way to get rid of metabolic waste, and they will unfortunately grow and continue to spread through clonal expansion in that distant site. So I hope now we have a clear understanding of the differences between invasion and metastasis. Thank you very much for watching, and thank you for your patience and your kind consideration. And please follow and like and subscribe, and see you in our next lecture. Thank you very much.